When it comes to chomping down on our favourite foods, the teeth are where the rubber meets the road. For that reason, the nature of tooth surfaces can tell us a lot about what different animals eat and how they eat it. The patterns of shapes across the functional surface of teeth are known as dental topography, and determining trends in these patterns can help us work out how animals live in and impact their environments. So how do we study dental topography then? In this video, we're going to go over the basics of the most common ways we study tooth surfaces and what these methods can tell us about animal biology. Dr. Rex here. Welcome to the Scullywag Lab, where I break down the bare bones basics of skull science. You know, people have been looking at the teeth of animals for centuries now to try to recognize patterns between tooth structure and diet. At first, this involved basic descriptions and simple measurements. While some of these continue to be used today, there are some new methods that have brought dental analyses into the digital age. This can include shape analysis using geometric morphometrics, but you can see that process in a previous video I made that breaks down that method. In this video, I'm going to break down some other methods you might come across in research papers. This includes relief index, orientation patch count, and Dirichlet normal energy. So let's get into it. Let's start with the simplest of these, relief index, often shortened to RFI. This effectively measures how much height and steepness a tooth packs into its two-dimensional footprint. It's represented by the relationship between the 3D surface area of the tooth crown and the 2D area of viewing it from above. Looking at this monkey tooth, this is a 3D surface of a tooth crown with the edge determined by where the gums used to be. So the total area of this 3D outer surface is the 3D surface area. Now, if we scale the vertical axes to 0, we get a flattened projection of the tooth's upper surface. This represents the 2D view from above. In earlier research, the 3D area was simply divided by the 2D area to give a basic relief index. More modern studies use a modified version of RFI though, that log transforms the relationship. Using this approach, a larger RFI usually means a tooth has taller cusps and steeper surfaces. We often see this in animals that need sharper crests for slicing through leaves or cutting into insects or slicing through other tough foods. Alright, next up, let's look at orientation patch count, or OPC for short. By measuring how often different parts of the tooth surface are facing the same directions, we can get an idea of how complex the tooth surface is. In Blender, I've visualized a kind of OPC by separating 360 degrees of orientation into 8 primary directions, each representing about 45 degrees with a different color. As it happens, while working this out, I accidentally created this oil stain texture. Anyway, notice how different parts of the tooth surface have the same colors? Well, these are the parts of the tooth that are facing the same general direction. Counting how many of these isolated patches of different colors there are gives us an orientation patch count. Now imagine if we had a really simple tooth, like a canine or something. This would have very few patches, maybe even just one patch per color. This would indicate a low surface complexity. But a molar with more complex grinding or shearing surface will generally have a higher patch count. In this pretty well-known paper, carnivores and herbivores could be differentiated based on patch counts across different groups of mammals, in this case carnivorans and rodents, so quite different, with herbivores generally having more complex teeth for better processing plant material. Some really nice results there. Alright, now let's get into a more complex measure of dental topography, Dirichlet normal energy, or DNE for short. Now to understand this method, you need to know what a normal is. The digital meshes we use for analysis from scanned specimens are made up of tiny simple shapes. These are most often 3-cornered triangles or 4-cornered quads. Each of these shapes is facing a specific direction, and that direction is represented by a line perpendicular to the surface called a normal. Using our trusty mesh from earlier, I've instanced a normal vector line for each little triangle. Notice how when the surface is flat, all the normal vectors are pretty much doing the same thing. But when there's a sharp edge, their directions rapidly diverge in different directions. Well, Dirichlet normal energy is basically a measure of how rapidly these orientations can change across a tooth surface. Flatter, less complex surfaces will have low DNE, usually a greenish color on these maps, while higher energy regions, like cutting edges for example, will move into yellow or even red colours, while the bluer areas tend to be more of the pitted or concave parts. This can be used to infer different feeding behaviours. For example, high D&E is often associated with sharper cutting surfaces that might be used for shearing flesh or piercing insect exoskeletons, while lower D&E tends to occur on smoother surfaces that might be better for crushing. So there you have it, that's the lightning fast basics of the most common ways we look at variation in tooth surface topography. In essence, RFI measures relief, OPC measures complexity, and D&E measures sharpness and surface variability in general. These three methods have been used to relate feeding ecology to dental anatomy and can be applied to all kinds of different animals, both living and extinct. In fact, I just used D&E to look at feeding ecology in 400 million year old placoderm fish jaws, but that's for another video. If you enjoyed this content, leave a like and subscribe, and I'll catch you in the next video.